Hello, Tam here. Welcome to lesson 2. Today we're going to be doing the Biuret demo and talking a bit more about protein structure. So we're covering specification point J here. Okay, so the learning objectives for today are to be able to recall and draw the condensation and hydrolysis reactions between amino acids, to understand how the Biuret reagent can be used to test for the presence of protein, and we'll be covering your first practical assessment today, which is CPAC 5, and then to describe and compare the structure of globular and fibrous proteins, explaining how their structure links to their function. So please pause the video and complete activity 1. Okay, and the answers for this. So the first one, explain the difference between qualitative and quantitative. Well, a qualitative measure is basically a descriptive measure. So for example, describing what a colour change looks like, the colour changed from green to pink or whatever. Quantitative is giving a numerical value for this. What test detects the presence of protein? So that's the Biuret test. Again, that's what we're going to be doing today. Suggest a situation when detecting protein in the solution may be necessary. So this would be, for example, if you're trying to measure for medical reasons the presence of protein in urine to detect kidney damage. Or maybe it's in food testing, so to test for the presence of protein in particular food samples. You should have an image that looks like this for your condensation and hydrolysis reactions. Just one thing to note here is that in my image, the residual groups aren't written as R, but I've actually given a specific amino acid, but you guys should just have an R in those points. So what do we need to remember when drawing graphs in biology? There's a few things to remember here. The first thing is always use a sharp pencil. This helps you plot accurately. Plot at least half of your A4 graph paper. Now this doesn't just mean extending your axes to make them bigger, but this is your plot where you've got your crosses and your lines. So make sure that is at least half of your A4 paper. Use appropriate and linear scales. So remember, a linear scale is where you've got the same gap between each of the values that you've put on your graph axes. That you have a value at the X and also the Y origin. So here I'm going to lose a mark on this graph because I've only got one value for the X and Y origins, but I would need 2. So I'd need one here and I'd also need one here. And they don't have to be 0. So my Y-axis might start at 10 and my X-axis might start at -1, for example. The X and Y axis titles should correspond to your table. Now this is a really important thing. Please check that whatever is in your table and whatever you're plotting corresponds to the axes on your graph. So if you're plotting the mean concentration of something, for example, it should say along that axis mean concentration of whatever. And then accurate plotting. So again, this links back to using a sharp pencil. You'd be surprised how many people lose marks by having a non-sharp pencil. So other errors in the graph that we maybe could be able to spot. So you might have seen that we've only got like one value on the y-axis, but we're going to want to put more than that, especially if we're plotting accurately. Okay, and that will show us our linear scale. We've also not got crosses on here, so we'd want to be putting crosses or dots at the exact points that we're plotting. In biology, we should have straight lines between our plots. Okay, so we should be using a ruler to draw straight lines between them. Here you can see we've got a curved line. Here we've lost marks again, as we said, because we've only got one origin, so we should have 2. And also we've plotted above what our scale says. So we've only put 40 on the x-axis as the top number, but actually our plot should be within the values that we give. Okay, so we should have another number up here. Okay. So your teacher will be projecting the Biuret demo on your Microsoft Teams account. So please access that and watch their video overview of it, and we'll talk a little bit more about it in the next few slides. So you should have completed your practical independent study for today's lesson. So just to review some of the questions down here. How do you zero the colorimeter? Now your teacher's actually going to show you this in their demo on Teams, but basically to zero the colorimeter, you're going to press the R button. Now that's going to reset it, and you're going to zero it using a particular solution. So in this case here, we're going to zero it with our 0 grams per decimeter cubed of albumin solution. Okay, now whatever you zero it with will vary depending on the practical. The filter that you're using, so today we're going to be using a 550 nanometer, or it's a yellow-green filter, so it'll be shining a yellow-green light through our colorimeter. We'll come back to how the colorimeter works in a second. Are we using absorbance or percentage transmission? So we're going to be using absorbance, but you can see on the colorimeter there's a little button here which says abs/percentage trans. Now that basically means that you can change the units. You can see on my colorimeter at the moment it says abs, so it's measuring in absorbance. Describe the colour change that you expect to see if the protein is present in a sample. So in the Biuret test, the solution will turn from blue to purple in the presence of protein. So remember, you should say the colour at the start and also at the end. So just a couple of things to review with you before you watch your teacher demo. So the first thing is, how does the colorimeter work? Now basically the colorimeter is measuring or giving a quantitative value for the colour change that you're going to see in your Biuret reagent. So here, this is what the Biuret reagent will look like at the start. It's a blue colour, and if protein is present, then that colour will change to purple. So I remember this by P for protein and P for purple. So here you can see the colour change is purple. So obviously there's going to be a bit of a scale. It's not the blue Biuret reagent isn't just going to turn to one colour purple. Depending on how much protein is there, you're going to get a more or less intense colour change. So we can get a quantitative value again for this using our colorimeter. Now how the colorimeter will work is that a light will be shone through the solution. You can see the solution is here in a little cuvette, so just there, in a tiny little square tube, and we put in our solution into the colorimeter, and then we shine the light through it to get our absorbance reading. Now we're working with 550 nanometers today, which is basically a yellow-green filter, so it's a yellow-green light. And we're using that one because the solution is turning blue to purple. So we can't really shine a blue or purple light at it, because otherwise, depending on the colour of the solution, it's just going to reflect it straight back. So we're going to use a colour that's a different wavelength from those. And so we shine the light through that solution, and there's a little digital strip that will measure how much light gets through it. And in this case, it's going to tell us how much is absorbed by the solution. Okay, but again, we could measure in percentage transmission and get a value out of 100 for how much light gets through the solution. We're also going to be working with egg albumin protein today. So what we need to do in this practical, first of all, is to generate a calibration curve. Now to do that, we have to mix a range of different protein concentrations. So in your independent study, you had this table here, and this showed the different volumes of albumin, which is protein, to water that we'd need to mix to get these concentrations. Okay, so you might have identified that in order to get a 2 grams per decimeter cubed dilution, we mix 5 centimeters cubed of albumin with no water. Okay, so that would give us our strongest concentration of protein solution, and we'd mix the different volumes until we got one at the very other end that had no protein in, so 0 grams per decimeter cubed of protein. What we will also find here is when we mix the Biuret reagent with these, that we get a variety of different colour changes, ranging from blue, which we'll see, which we'll see in this tube here, all the way down to deep purple, which we'll see in this tube here. And then your teachers are again going to show you this in the demo, but what we do is we take a sample from each of these and we put it into the colorimeter, okay, and we press the T button. So we've already zeroed it with our 0 grams per decimeter cubed of albumin. So now we're just going to test it. And what it's going to do is test and compare the colour of the new solution compared to the one that we set the colorimeter to. So we'll get our absorbance readings and we can insert them into this table here. So your teacher will, in the demo, generate some results for the absorbance values at 550 nanometers on the colorimeter for each of these protein concentrations. And what you guys will do is, using the graph paper that you got given last lesson or that in the activity sheet, plot a calibration curve. Now here's mine on the board. Please excuse the fact that it's a computer generated one. You guys need to plot one by hand. And what you can notice here is that as the concentration of protein increases, the absorbance also increases. So basically, as there's more protein in my solution, the Biuret reagent changes to purple or a deeper purple colour. So now we can use this graph to basically find out the concentration of protein in a sample that is unknown. So maybe, for example, I might be a doctor and I need to test how much protein is in someone's urine to see if they've got kidney disease. So I've got their sample and I put it into the colorimeter and maybe I get an absorbance reading of 0.1, okay, 0.1 abs. So what I'm going to do now is I'm going to read off my graph, and I'll do this with a ruler. Please excuse my wobbly line for now. Okay, so you guys will do this with a ruler, but I'm going to read off from 0.1 abs, and then I can, again with a ruler, read down from there, and it will give me the value or the concentration of protein in that solution. So that's what you guys will need to do, is to find out the concentration of protein in the unknown solution. You'll need to generate a lab book on Teams. There's a link in your activity to help you do this. And your lab book page should look something like this. Okay, so again, the results table should be in the independent study for you to copy and paste over and then to fill in. The graph you're going to hand draw, and then you're going to take a photo using OneDrive and upload that onto your OneNote lab book. The instructions are all in the activity, and there's also a video link to help you with this. So activity 3, I'd like you to pause the video and to try some of those questions. Okay, so questions here then. The results that we'd expect to see if protein was present in a Biuret test. Now the key thing here, or top exam tip, is remember to say the start and end colour in the reaction. So for example, if protein is present, then you will have a colour change from blue to purple. Now we have to say what the colour at the start was, because otherwise if we just say that the colour was purple, it might have been purple at the start anyway. So make sure you say the colour change. If protein is not present, the reagent would remain blue. So again, that indicates what it started and ended as. Another question is, the mammal's urine was tested for the presence of protein. The concentration was found to be 0.16 grams per decimeter cubed. Write a conclusion about the health of the mammal. So a healthy mammal should have a concentration of 0 grams per decimeter cubed of protein in the urine. And this is because in the kidneys, you've got a structure called the glomerulus. And if the glomerulus becomes damaged, then it can affect the molecular sieve, which you'll learn more about next year. And it just basically means that proteins leak out of your blood and into your urine, and that shouldn't happen. So it would imply kidney disease if you have some protein in your urine. So what are globular and fibrous proteins? Now, just as a quick overview, basically there are different shapes and functions of proteins. So globular are 3D in structure in terms of they're more sort of spherical in structure, and these would be things like enzymes that have a specific 3D shape that helps it do its function. A fibrous protein, so these are generally long and thin and strands of protein, and these help with structural support. So for example, something like collagen would be a good example of a fibrous protein. Please pause the video and complete activity 4. Okay, so the answers for activity 4, we should have in the table a few things that like this. So globular proteins, these can be soluble in water and also have a three-dimensional structure, for example, like enzymes. The function, so if it was something like an enzyme, then it might catalyze reactions, or they might be transport proteins embedded in the membrane that help substances get from one side of the membrane to the other. And again, shape is really important there because they'll be specific for whatever, a specific shape for whatever they're transporting. Some examples, as we said, so enzymes, channel proteins, there's lots and lots of globular proteins that we'll talk about throughout your course, things like hemoglobin as well. Fibrous proteins. So fibrous proteins are insoluble in water. They're long parallel polypeptide chains joined together, and they provide structural support. So this might be something like keratin or collagen. A couple of examples here. So collagen was one example we just gave, and here is a nice diagram of what collagen would look like. So you can see actually collagen fibers are made up of lots and lots of chains of these proteins. And here you'll notice it says alpha chains. So if you remember, we spoke about secondary structure last lesson, and one of the secondary structures is the alpha helix, and the alpha helix is a helix or coil of your polypeptide chain. So here in the collagen, you can see that the alpha helices are wound around each other. And you'll notice actually that there's 3 that are wound around each other in this collagen molecule. Hemoglobin is an example of a globular protein. So hemoglobin, you can see, has a specific 3-dimensional shape. And in this case here, actually, you might notice or have a think about what level of protein structure. So hemoglobin is actually a quaternary level protein structure because it's made up of 4 polypeptide chains. Your final question was to describe how globular and fibrous proteins are formed and explain the difference. You might want to just pause the video here and check your answers. Just take note of any keywords in bold, see if you can add them into your notes. And finally, the learning objectives that we've looked at today. So just remind yourself, see if there's any that you don't feel confident on, write down any questions to take to your next lesson. Okay, thank you.